Hi everyone, here we are for Chapter 6, Part A. So Chapter 6 is all about blood, and in Chapter 5 we looked at the cardiovascular system and we looked at blood vessels and how the heart worked, and now we're going to look at the blood that flows through them. So you'll notice functionally there's a lot of similarities that were stated for the whole system, and then you'll also kind of see where a lot of our terms will overlap, and at this point I expect that you know the parts of the heart, you know the differences of the blood vessels and the flow, so we're moving forward with that base knowledge. So the functions of blood can largely be divided into transportation, defense, and regulatory. So in transportation we could divide that into four, right? Or five. It transports oxygen, it transports nutrients, blood transports wastes, it transports carbon dioxide, and it transports hormones. So those could all even be divided. Transportation, of course, of oxygen and nutrients and hormones will be to cells, and then the transportation of wastes and CO2 will be away from the cells in your tissues. Defense-wise, blood is carrying white blood cells, and white blood cells are the cells that are going to fight infection. So part of the blood's functions overlaps with the immune system, in that the immune system is largely made up of white blood cells, although there's some other things too. And then the third category are regulatory functions. So blood helps regulate your body's pH, it helps regulate the body's temperature, it helps to regulate your body's water balance and your salt balance, and so it's very helpful at maintaining homeostasis. So blood's composition is pretty much divided into two major categories: formed elements and plasma. So formed elements are things that I suppose you and I could maybe squish with our fingers. And yes, they're microscopic, but they do have a structure. They have some sort of membrane or membrane-like thing surrounding it, versus the plasma is just water and dissolved salts. So formed elements, there are three types: red blood cells, white blood cells, and platelets. Red blood cells, RBCs, are also known as erythrocytes, and white blood cells, WBCs, are also known as leukocytes. And I do want you to know both of those, mainly because by recognizing them, it'll help you recognize when other terms relate to them. So leukemia is cancer of the leukocytes, and erythropoietin is a hormone that's going to influence the production of erythrocytes. Think of erythro meaning red in Latin. That might help some folks, and leuko meaning white, and that cyte again meaning cell. Platelets are not an actual cell. They're a little tiny piece of a cell, as we'll see later, but all three of these are produced in the red bone marrow. And red bone marrow is inside your bones, and you have more of it as a kid, and then as you get older, you have less and less of it. So those things are all in blood, and then plasma makes up the rest of the blood. And plasma is, mostly water, and then the remaining 9% are salts, which divide up into ions, and other organic molecules. So think of plasma proteins. There's going to be a little glucose in there. There could be some lipids in there too, but typically it's the proteins. And there's three big types of plasma proteins that might sound familiar to you, or if not, they will sound familiar later. So albumins are a protein that help maintain osmotic pressure and help with transporting items. So maintaining osmotic pressure, just by being present, it's preventing your plasma from being almost pure water. Fibrinogen is another plasma protein that is going to help in the formation of blood clots. We're going to see that come up later. And then globulins are useful again for transporting things throughout the blood. So don't worry about memorizing this whole image, but I just wanted to point out these formed elements. So again, thinking back to our red blood cells, white blood cells, and platelets coming from red bone marrow. And they all come from this generic stem cell. A stem cell is just a cell that hasn't specified yet. It's still just really generic. And so what we see is that it can divide and divide, and we have the red blood cells that come from that. We see then in the middle are 5 types of white blood cells: lymphocytes, monocytes, neutrophils, eosinophils, and basophils. All 5 of those are white blood cells or leukocytes. And then at the far right we see the platelets, and the platelets, notice size-wise, are a lot smaller than all of the cells to the left. So technically they could be called thrombocytes, and if you want to learn that, that's perfectly fine, but I'll just refer to them as platelets. And platelets, basically what happens with platelets is a megakaryoblast, or megakaryocyte, it gets really big, and it starts branching off into the blood vessels, and little pieces of it breaks off and those are the platelets. So we're gonna, in part A here, we're gonna now focus just on the red blood cells, or the erythrocytes. Part B will get into leukocytes and platelets, and part C will then look at blood typing. So red blood cells have no nucleus. They have very few organelles, so they're basically just jam-packed full of hemoglobin. That's pretty much what's in there. Shape-wise, they have a biconcave shape. So that's what when we think of, it's almost a donut, except that it's not an actual hole in the middle. It is, it's just as if you had pinched down on it, right, with your thumb and your forefinger, so it's very thin. So that biconcave shape increases the amount of surface area that the red blood cell has, and that's going to be really useful for gas exchange. The more surface area you have, the more oxygen can come in and out, and the more CO2 that can come in and out, and that's going to help blood transport a lot of gases. So if the red blood cell doesn't have a nucleus and it doesn't have very many organelles, right, it's basically just full of cytosol. So what's sitting in that cytosol, in that cytoplasm? And basically it's just full of hemoglobin. Every red blood cell contains about 280 million hemoglobin, and all of those hemoglobin can bind 3 oxygen molecules. So you can have lots and lots of oxygen in each red blood cell. If your oxygen levels in the body fall to low levels, the body has something called chemoreceptors that can sense that, because it's sensing a chemical. And so what that does is then stimulates your kidneys to release the hormone erythropoietin, and there's that erythro that we mentioned earlier. For short, it's EPO, but be able to recognize both names. So erythropoietin is a hormone that goes through the blood, and when it gets to the red bone marrow, it stimulates the red bone marrow to produce more erythrocytes. Now, this isn't always going to be perfect, because maybe you have plenty of red blood cells, but the hemoglobin isn't working, or there's a malformation that's preventing the oxygen from leaving the red blood cell. So it's not a perfect solution, but the body's like, hey, oxygen's low, I need more red blood cells. That's kind of its initial go-to. So because red blood cells don't have a nucleus, there's no point in them dividing, because they have no genetic information to pass on. They don't even have the genetic information to conduct the process of dividing. So basically, once they're produced, they just have a lifespan of about 128 days. Every time a red blood cell goes through a capillary, it has to bend in half, and it actually, I mean, it's not a super rough exterior or interior of the capillary, but you know, it does get a little wear and tear essentially. So after 120 days, it's a little bit rough around the edges, literally. And so in the liver and the spleen, as blood passes through those organs, they will clean out and just basically destroy and recycle, for the most part, many parts of the red blood cells. So we want to look at how carbon dioxide is transported by the body. So for review from the previous slide, oxygen is transported by binding to hemoglobin, but CO2 can do one of 3 things. CO2, a little bit of it, will just dissolve in the plasma, and it will stay dissolved in the plasma as CO2, and it just travels around. About a quarter of the carbon dioxide will actually bind to hemoglobin in place of oxygen. And it actually binds to hemoglobin a little bit tighter than oxygen does, so that's why we don't want to be exposed to too much CO2 at once. It'll block out the oxygen and your tissues won't get the oxygen they need. But it just normally 25% bound to hemoglobin is totally normal, totally healthy. The remainder of it, so most of the carbon dioxide, about 68%, will be transported through the body as a bicarbonate ion. And that's due to this equation that you see here, which is not essential for you to memorize, but it is a very useful equation that you'll see repeatedly in the human body and in just biology. So it's the equation that when we add carbon dioxide and water together, they make a weak acid called carbonic acid. And that carbonic acid, pretty much as soon as it's made, will split again into a hydrogen ion and a bicarbonate ion. And this will go both directions. You'll notice the arrows here are going to the left and to the right at the exact same rate. So the body is constantly fluctuating between these and finding a balance. But that means that these bicarbonate ions are going to hold most of that carbon dioxide, will actually be part of a bicarbonate ion as it moves through the blood. Okay, so those are what red blood cells are. It's how they're shaped, how that shape affects their function of gas exchange and carrying oxygen. And now we're going to look at people saying, hey, I understand how red blood cells work. Maybe I can try to enhance my athletic performance by adding more red blood cells so that I have more oxygen to my tissues and I can perform better. So this is the idea behind blood doping. The idea was to increase the number of red blood cells, therefore we could increase athletic performance because blood could carry more oxygen and therefore reduce fatigue, just because we had more hemoglobin in the red blood cells. So what folks do when they're using this method is you inject someone with EPO, remember that erythropoietin hormone, and they do this months prior to your athletic event. And what that does is that it increases, of course, the production of red blood cells. They would then remove some of the blood from the body and take out the red blood cells, and then they would actually store them either in a freezer or a fridge, or in appropriate conditions so that the red blood cells are not harmed, and they get stored, and then just a few days before the athletic event, your own red blood cells are added back into your blood. So you have now more red blood cells in your blood, therefore more hemoglobin, therefore more oxygen. But what initially people didn't account for, and people who don't actually know about it but try to use it, is that because you have more red blood cells, it actually makes your blood a lot thicker. Because a red blood cell, even though we need a microscope to see it, blood-wise is a pretty significant cell. I mean, it is an entire cell, and capillaries are super, super, super tiny. They hardly even fit one red blood cell. The red blood cell has to fold to fit through it. And if you just have so many red blood cells, then that's going to make it a lot more likely that you're going to get a clot made out of those red blood cells, and they're going to back up and could lead to a heart attack or a stroke, and of course death could be the end result of that. So blood doping was an interesting application of biology, but unfortunately it actually is not a healthy option. A few red blood cell disorders that can occur in the body include anemia. There are tons and tons of types of anemia, but essentially as a group of diseases, anemia is when there's too few red blood cells or there's too little hemoglobin in your red blood cells, and you just can't transport enough oxygen. So as a result, you feel very tired, you feel very run down pretty much all the time. Sickle cell anemia is one particular form of anemia where the red blood cells are sickle-shaped. They look like these images on the left. They're not the nice round donut shape that we're accustomed to. And because of that shape, they're not able to fold very well when they go through those capillaries, like we mentioned the normal red blood cells could. And so as a result, it's much more likely that they'll rupture, and when they rupture, their hemoglobin is just released into the blood, and basically at that point it's not even useful. It's not able to carry oxygen unless it's in a red blood cell. So that's what's going on with sickle cell anemia. With hemolytic disease of the newborn, it's kind of a mouthful for a name, but what's going on is that red blood cells in the baby are going to be rupturing before birth and continuing after birth. So sometimes this can even lead to a miscarriage if not treated. So we'll talk about this more at the end, but what's going on is a mismatch between the mom's blood type and the baby's blood type. And that's completely normal to have different ones, because when a baby is made, of course, we're mixing half of the genes from mom and half of them from dad, and that can result in a different blood type. But what that means is that if we see that difference in positive and negative blood, it could lead to some damage in a baby. So more coming at the end of Part C, but we just want to remember that this is a disease of the red blood cells.